Polypeptide synthesis by Adam Downey. Polypeptide synthesis is the biological production of peptides. Amino acids are conjoined together through peptide bonds, which fold into the form of a protein. We require the process of polypeptide synthesis because proteins are essential for cell and bodily functions. Codes for certain proteins are kept tightly packed in a DNA strand within the nucleus. Polypeptide synthesis, however, can only occur in a ribosome in the cytoplasm. So how do we get the information from the DNA out of the nucleus? This is the first process of polypeptide synthesis, transcription. In the process of transcription, DNA is transfigured into mRNA, messenger RNA. Transcription has 3 steps: initiation, elongation, and termination. In initiation, RNA polymerase binds to the promoter section of the DNA. Elongation is the stage where the RNA polymerase reads the DNA strand and begins adding the complementary base pairs to the mRNA strand. For a base pair to be complementary, they must match. A always pairs with T, and G always pairs with C. RNA is very similar, except it does not produce thymine, instead uracil, so A matches with U. Termination is the final stage of transcription. Termination indicates to the mRNA strand that it has reached the end of the coding region. Once this has happened, the RNA polymerase will be released from the template strand. The strand will then go through processing, removing introns through RNA splicing and exons to synthesize proteins. Translation is the second phase of polypeptide synthesis. The main idea of this process is to convert the mRNA into a functional protein. The mRNA is now in the cytoplasm and attached to a ribosome. The ribosome is what converts the mRNA into a protein and is composed of RNA, ribosomal RNA. tRNA, transfer RNA, can be found in the cytoplasm with an amino acid attached to them, remembering that amino acids are considered the building blocks of proteins. It is the tRNA that will construct those amino acids together in order to create a protein. The mRNA is going to instruct which amino acids from the tRNA will be required. Once the tRNA has found their complementary base, they are going to transfer the amino acid and eventually leave, leaving the amino acid behind. This repeats and they attach themselves, forming a peptide chain. Let's take a look at the following example of translation. Here we have a 12-base strand of mRNA which has been transcribed and spliced in the nucleus and has now bound to a ribosome in the cytoplasm. The order of nucleotides in the mRNA strand are AUG, CUA, AGU, UGA. As explained before, a tRNA with the corresponding anticodon will bind to a codon on the mRNA, bringing in the first amino acid to our chain, which is methionine, which we can see from our codon wheel. Now that methionine has started our polypeptide chain, the ribosome moves on to the next codon on the mRNA, which is CUA. The tRNA with the corresponding amino acid, leucine, will be brought into our polypeptide chain. We now move on to the next codon, which is AGU, which codes for the amino acid serine. Finally, we reach our last codon, UGA. As you can see from the codon wheel, UGA is a stop codon, hence the translation process is terminated and we are left with a polypeptide chain with 3 amino acids. This is a functional protein as it has the right amino acids as coded by the DNA. Yay! Now let's talk about how protein synthesis can go wrong, specifically when we have a point mutation. The effect of point mutations can vary depending on which nucleotide is affected. There are 3 potential effects of point mutations, specifically silent, missense, and nonsense mutations. Silent mutations are when a point mutation in the DNA strand coincidentally leads to the creation of the exact same amino acid, meaning there is no effect on the protein structure or function, hence why it is called a silent mutation. For example, if we had TCA in our DNA template strand, which would have produced an mRNA strand with AGU, but a point mutation occurred, which changed the A to a G in our template strand, so we now have TCG instead of TCA. and the mRNA now has AGC. The amino acid in our final polypeptide chain will still be serine, but we look at the codon wheel.
So it is silent!
Nonsense mutations are when a point mutation in the DNA strand leads to a stop codon instead of the intended amino acid. For example, let's look at the segment of template DNA which has ACG. After transcription, our mRNA will have UGC, and after translation, we will have a cysteine amino acid in the polypeptide chain. Now imagine a point mutation occurred in our original DNA template strand, leading to the segment to now be ACT. After translation, we will have UGA, and if we look at the codon wheel, we can see that UGA codes for a stop codon. This will lead the ribosome to stop translation, and hence the protein will be missing essential amino acids and will not function properly. Missense mutations are what we are going to focus on more in depth. Missense mutations, in a nutshell, are when a point mutation leads to the incorrect amino acid being coded, leading to the protein being structurally and functionally wrong. Here we have a segment of double-stranded DNA, with the template strand being TAC, GAT, TCA, and ACT. This DNA strand has been unaffected by point mutations. This DNA strand will be used to create a protein, a polypeptide chain. As explained, the first step in protein synthesis is transcription, which is initiated by RNA polymerase. The RNA polymerase binds to the DNA strand, and because it can only create mRNA in the direction of 5' to 3', we'll use the template strand of the DNA which reads in the direction of 3' to 5'. The RNA polymerase binds to the template strand at the primer, then begins connecting the corresponding nucleotides, specifically AUG, CUA, AGU, UGA. Once the RNA is complete, it undergoes splicing, then is ready to exit the nucleus through a nuclear pore. Now we're in the cytoplasm, where the mRNA binds to a ribosome. As the ribosome reads the mRNA, it organizes tRNA molecules according to the codon-anticodon pairs as shown. This leads to the arrangement of amino acids, which we can predict from the codon wheel. The codon AUG codes for methionine, the codon CUA codes for lysine, the codon AGU codes for serine, and the codon UGA codes for a stop codon, terminating translation. We are now left with a 3-amino acid chain which is structurally and functionally correct, because the original DNA was unaffected by point mutations. Yay! Now let's look at what happens when we have a substitution point mutation which leads to a missense mutation. Here we have an identical double-stranded DNA as seen before, however there is a slight difference. A substitution point mutation has occurred on one of the adenines in the template strand, leading to a thymine to be present instead. Point mutations can arise through various problems, for instance, radiation or chemical mutagens. Though they seem small, we will see how this can affect the entire protein function. As before, the DNA strand will be used to create a polypeptide chain, and the first step in protein synthesis is transcription. The RNA polymerase binds to the template DNA strand, then begins constructing the mRNA strand. However, there is a slight change in the mRNA now. Our new mRNA strand is AUG, CUA, AGA, UGA. Though the change is small, there is now an adenine in the place of what should have been a uracil on our mRNA. For the moment, this has no effect, and the mRNA is spliced and sent out of the nucleus for translation. Now we're in the cytoplasm, and as before, the mRNA binds to the ribosome. The ribosome brings in the corresponding tRNA molecules, and we can use the codon wheel to see that the amino acids will be arranged. The codon AUG codes for methionine, and the codon CUA codes for leucine. This is where things get weird. Our third codon, AGA, which should be AGU, now brings in an arginine amino acid instead of serine amino acid. Then UGA codes for a stop codon, terminating translation. We are now left with a 3 amino acid chain, but as you can see, the third amino acid in the sequence is now an arginine instead of a serine. Because proteins have highly specific structures, and even one wrong amino acid in its polypeptide sequence can disrupt structure, hence function, this change means the protein cannot function properly. This is what missense mutations are. Point mutations in the DNA lead to a different amino acid in the final polypeptide chain, leading to a dysfunctional or non-functional protein, which is very not good.